pleasure to introduce Dr. Chantal McCabe. She has been in our department for a while now. She got her bachelor's degree at University of Minnesota in Duluth and then came here to work with Drs. Graham and Stanzo for her PhD program, where she worked in soybean, and has now graduated and moved into a postdoc position here, also working now for Dr. Graham, and she is here to talk about her research that she's been doing. I do want to congratulate her. She received one of the NIFA postdoctoral, not scholarship, fellowships. So she will be doing some additional work on that, and without further ado, let's give Chantal your attention. Can you guys hear her okay back there?
Is this on? Good?
Okay.
Okay. Well, today I'm going to be talking about some work that I've done characterizing the RBS3 locus in soybean. As we know, soybean is a really economically important crop. But every year, we lose a lot of yield due to pests and pathogens. And here's a list of some of the most devastating diseases in soybean, ranked by the number of bushels lost. And today, we're going to focus on brown stem rot. And every year, there are around 13 million bushels of soybean lost just due to this disease. And this number probably should be a lot higher because we have problems phenotyping and identifying this disease. Phialophora gregata is a fungal pathogen that causes brown stem rot. This is a soil-borne fungus. It causes foliar symptoms and stem symptoms. The foliar symptoms appear as leaf necrosis and leaf chlorosis. Now these look almost identical to the foliar symptoms produced by sudden death syndrome and iron deficiency chlorosis, which are both also really prevalent in the Midwest and infect soybean. So in order to identify brown stem rot properly, you must cut the stems open. So this involves splitting the stem open lengthwise and looking for the brown, dead, and discolored tissue inside the stem. A susceptible plant will have the brown, dead, and rotten tissue, just like the name brown stem rot would imply. But this phenotyping can't occur until 5 weeks after infection, so it's a very long process. Takes many weeks to phenotype, you need multiple measurements, and splitting the stems open is a destructive measurement. You're killing the plant. So phenotyping is really difficult. There are 3 resistant genes called RBS1, RBS2, and RBS3. These were all identified in the 1980s by allelism studies. So these studies suggest that they're independent dominant genes. A few years later, these 3 were mapped to chromosome 16 in soybean. So this diagram is a region of chromosome 16. We have the markers here on the left, the position, these numbers are on the right, and RBS1 has been mapped to this long region between SAT215 all the way down to SAT431. So it's a 33 centimorgan long region. RBS2 has been mapped right here between SAT244 and SAT431. It's 20 centimorgans and completely overlapped by RBS1. RBS3 is the finest mapped right here, K375 to SAT244. Again, it's completely overlapped by RBS1, but it's the finest mapped at 5 centimorgans. So if you're a breeder and you want to integrate one of these genes into your elite line, or pyramid resistance and stack 2 of them into your elite line, it's a really difficult task. How do you even begin? It's a really complicated and confusing region. And if it wasn't as confusing anyways, there were 2 studies recently published by Rinkner in 2016. One was a GWAS study and the other was a fine mapping study. And he suggested, based on the results of these 2 studies combined, that all BSI resistance is actually conferred by one single gene, and this gene is located here at the star. So it's a very complicated and very confusing region. So we have problems phenotyping for brown stem rot and problems genotyping for brown stem rot. This RBS region is not well defined. The traditional map-based approaches have not been successful in characterizing resistance. So we need something different, an approach that combines both breeding and genomic technologies to really understand BSI resistance. So in my study, I had 3 objectives. The first is to characterize RBS3 resistance genes. The second is to develop molecular markers that can be useful and improve marker-assisted selection. And the third is to identify downstream defense gene networks. So in my RNA-Seq study, I had 2 genotypes, resistant and susceptible. The resistant genotype is PI437970, which has the RBS3 gene. And this is the gene that I chose because it's the finest mapped. It's the one that's only 5 centimorgans long. The susceptible genotype I used was Corsoy79. And in each genotype, I infected half the plants with the pathogen, P. gregata, and half with water agar as a mock inoculated control. And both treatments were injected into the stem of the soybean at the soil line. And then 1 week after infection, I collected leaf, stem, and root tissue from the plants. And then this RNA was sequenced, and I went through the bioinformatic pipeline and looked for genes that were differentially expressed in response to genotype, in response to treatment, and the genotype by treatment interaction effect. So in response to genotype, differences between the resistant and susceptible genotype, regardless of infection, there were 6,938 genes significantly differentially expressed. Now, the 2 lines that I used were unrelated, so we expect a lot of differences. And most of these differences probably are not related to defense. But because we know the location of RBS3, we were able to look at genes differentially expressed in that region. So in this RBS3 region on chromosome 16, there are 37 genes. The genes are listed in this heat map. 21 of those genes were significantly differentially expressed in my data set. This heat map is made based on fold change differences. So if the gene is blue, it's expressed more in the resistant genotype. If it's yellow, it's expressed more in the susceptible. And this is expression in all 3 tissues, the roots, the stems, and the leaves. And what stands out the most is this really dark blue region being expressed strongly in the resistant roots. These are 12 different receptor-like proteins, or RLPs, expressed very strongly in the resistant genotype. So this tells us, because they're expressed so strongly and there's so many of them, that RLPs are important in RBS3-mediated resistance. And because they're expressed in the roots, regardless of infection in the resistant genotype, we know that P. gregata resistance is not inducible. In the field, this pathogen enters the plants through the roots. So we would expect that those genes, the resistance genes, should always be turned on to recognize the first moment of pathogen infection. So this pathogen surveillance by these RLPs is always on. So RLPs are really important. So we wanted to look at the spread of the RLPs and other R genes in the whole RBS region. So this is a zoomed in picture of the figure a few slides ago. We still have the markers on the side, the position. We're showing half of RBS1, all of RBS2, RBS3's up here again, and then the ring or stars up above. So in that entire RBS region, which is 33 centimorgans, there are 687 genes in Williams 82, the reference genome. We characterize these genes using interproscan. 18 of them are NBS LRR R genes, and they're shown as a triangle. There's one on the bottom, and the rest are way up above at the top of RBS1. 2 of these genes are RLKs, receptor-like kinases. They're also way at the top of RBS1. But 107 of these genes are receptor-like proteins. And they're all of the circles that are basically spread in this entire region. So 107. To give you an idea of how many that is, the entire Arabidopsis genome only has 57 RLPs. We have 107 in this small region on 1 chromosome. So RLPs are really important in defense. An RLP is made up of several domains: a signal peptide domain, a B domain, an LRR domain, a transmembrane domain, and a cytoplasmic domain. There are some differences in RLPs based on the LRR domain, different length based on the number of repeats that these proteins have. But they also differ based on their B domain. RLPs are unique. They can function in either defense or development, depending on the number of cysteine pairs in that B domain. So if the B domain has 2 pairs of cysteine residues, it functions in defense. If it has 1 pair, it functions in development. So out of those 107 RLPs, we had the full-length sequence for 65 of the B domains. They're all listed here. We did an alignment of those, and all of these 65 RLPs have 2 pairs of cysteine residues, so they function in defense. And by doing our alignment, we noticed that there were slight differences. So we were able to subgroup these into 5 subgroups that we're calling B1 through B5. So now I'm adding another line, another column onto this map from before. So the color-coded circles are the 65 RLPs that we know the B domain for. So at the top, we have these blue and purple circles. Those are B domains with RLPs B1 and B2. These green ones have a B3 domain, and the ones on the bottom that are orange and yellow have a B4 and a B5 domain. So the RBS RLPs contain the classic hallmarks of disease resistance genes. We have the conserved resistance motifs, the 2 pairs of cysteine residues. They're clustered in the genome. Not only are they clustered on this one region of the chromosome, but they're also clustered based on their B domain. We have evidence of recombination, that there's insertions and deletions that we've seen in the B domains. So these RLPs are classic disease resistance genes. So now I'm adding 3 more columns onto this picture. We wanted to see what the expression of these genes was in my data set. So there's expression in the leaves, in the stems, and the final column is in the roots. So looking at my genotype differences, any of these circles that are green are expressed more in the resistant genotype. The circles that are tan are expressed more in the susceptible. So in the leaves, there are not very many RLPs significantly differentially expressed. In the stems, there are not very many either, and they're all induced more in the susceptible. But in the roots, we have a lot of RLPs, and a lot of them are induced in the resistant genotype. And in fact, a lot of these are at the historic RBS3 locus. So looking at this, there are 12 RLPs expressed more in the resistant genotype at that historic RBS3 locus. These are 12 candidate genes for RBS3 mediated resistance. And remember that Rinkner suggested that all BSR resistance was conferred by one gene at that star. But in my expression data, none of those genes are expressed in the resistant genotype. Those genes are actually associated with susceptibility. Genes that are associated with resistance all occurred at the historic RBS3 locus, not in the region of the star. So the results suggested by Rinkner likely are not true. Looking at this picture, there are other clusters of RLPs as well. The cluster of B1 and B2s at the top, and the cluster of B4s and B5s at the bottom. It's likely that these other clusters could be correlated with RBS1 and RBS2 mediated resistance as well. So RLPs are responsible for P gregata resistance at the RBS3 locus. We've identified 12 candidate genes here. So using this RNA-Seq data as well, we were able to answer our second objective, which is to identify SNPs at this RBS3 region. So looking at the differences in the genotype data in the RBS3 region, which extended up to include the area until the Rinkner star, there are 534 SNPs. And these SNPs map to 66 unique genes. And out of the 12 candidate genes I identified, I have SNPs for 5 of those. And because we found these SNPs with the data from 1 week after infection, we can use these SNPs to screen for the disease earlier, faster, and more accurately, because you can't even phenotype until at least 5 weeks after infection. We can also use these SNPs to characterize resistance and find map resistance further. So switching gears a little bit and looking at the treatment effect, differences in the infected versus mock infected, regardless if the genotype was infected or not, or regardless of the genotype. In this comparison, there were 3,788 genes significantly differentially expressed. So on this line, there are 3,788 lines going down the heat map. It's organized by tissue and treatment on the bottom and expression pattern on the side. This colored bar graph has grouped all of these genes into 7 different clusters, and all the genes in a cluster should be expressed similarly. And what stands out the most is what's happening in the resistant infected leaves. They have the strongest treatment response. And this is at 1 week after infection. So this would suggest that the resistant response has moved from the inoculation point in the stem up into the leaves within 1 week, which is much sooner than the phenotype would suggest. So to determine what these genes do, we used gene ontology, Go terms. And the genes in each cluster have a unique biological function. Many of the clusters are related to the defense response. There's a cluster related to photosynthesis, and one related to the DNA replication and gene silencing and cell proliferation. We also looked at transcription factors that are significantly differentially expressed in response to infection. There were 30 different transcription factor families differentially expressed, and we looked at the response in the resistant genotype in response to infection and the susceptible genotype in response to infection in all 3 tissues. So a circle that's red is induced, and a circle that's blue is repressed. And there are several transcription factor families that really stood out, like Werke, MIB, homeobox, beta helix, loop helix, and AP2 EREB families. And these transcription factor families are known to be important in the defense and abiotic stress responses. So looking at the treatment response and the genotype by treatment interaction effect, which I didn't show today, and looking at the Go terms and the transcription factors, we were able to identify what the hallmarks of BSR resistance are. They are defense, DNA replication, and iron homeostasis. So in conclusion, by combining the RNA-Seq analysis with the historical mapping data, we see that this RBS region is really complex. But we were able to identify clusters of RLPs associated with RBS1, RBS2, and RBS3 loci. We found 12 candidate genes for RBS3 mediated resistance. They're all RLPs with the B3 domain, and it's likely that there's other RLPs associated with RBS1 and 2 resistance as well. We found 534 SNPs in the RBS3 locus. These can be really useful for screening for the disease, as well as fine mapping the resistance as well. We know the gene networks associated with resistance include defense, DNA replication, and iron homeostasis. And these methods of combining the historical mapping data with the genes, with the expression data, can be really useful for characterizing those complex regions. So we can use these same methods for characterizing resistance in other soybean diseases, but also use it for other crops as well. So thank you for listening today, and thank you to my postdoc advisor, Michelle Graham, as well as the entire Graham, O'Rourke, Sionzio, and Whittam labs.
Any questions for Chantal?
If you'll repeat the question for
Okay.
the audio.
Very nice talk. And so I have a question about the downstream network analysis.
Mm-hmm.
Is still based on these 2 very different lines. Is that right?
Yep, the treatment effect we looked at to identify the downstream defense responses. Yep, so
Yeah.
2 different lines, but the so we were looking at infected versus mock infected was the comparison.
Okay, so it's the same line, but infected and non-infected was compared.
The
Well, I think my question is, how do you rule out the significant Go term enrichment was not due to the natural difference of these 2 lines instead of the gene that you're focusing on, the tree you're focusing on?
Yep. So we did a genotype comparison, which would have showed what was the difference between the 2 genotypes, regardless of infection. But the pictures I showed at the end to look at the downstream defense was looking at the treatment response. So the way we had the comparison is the resistant of or the infected of both genotypes divided by the susceptible of both, or mock infected of both genotypes. So lots of different, in the pipeline that we looked at to get these differentially expressed genes, there were lots of different comparisons that we used. Mm-hmm.
Thanks, Chantal, for your talk. I really like this idea of using those SNPs as markers for the resistance
Mm-hmm.
to do an early screening. Have you checked those 534 SNPs in the data that was published for the GWAS? I guess they use a diverse panel. Were you able to see some of those SNPs in that data, and then take a look on the phenotype of those lines? Have you kind of test those to validate if those markers that you are identifying could be used in this data?
Mm-hmm. I haven't compared these SNPs directly to the other paper's GWAS data, so I don't know that answer. This SNPs are not in the 3 candidate genes identified in the previous paper.
Yeah, but maybe you can see a correlation.
Mm-hmm.
Because if they did a GWAS, they will have the phenotypic data and the genotypic data, and then you can also check in that those SNPs to check if there is some correlation
Mhm.
with resistance or not.
Thank you, Chantal. That was a nice presentation. How did you arrive at a conclusion that it's iron homeostasis, not other metal?
So the question is about iron homeostasis, and I didn't show a lot of that data here, but our lab has done a lot of work on iron deficiency sclerosis in soybean, and the results from those RNA-Seq studies and the results from mine are very similar, and they do produce a very similar phenotype. Some of the GO terms which are identified in the other analyses that I didn't show were specifically iron homeostasis, and a lot of the conical iron genes were differentially expressed in my data as well. Yeah, Shanmi.
Yeah, in the first set of papers, when they discovered resistant gene, those original study conducted with different sources of the resistant germplasm, or are they actually combined? Sometimes have one experiment with, let's say, one susceptible, one resistant with 2 QQ peaks. Because when you did your experiment, you say this one was RBS3.
Mm-hmm.
But then after you've done your study, you suspect those ILP gene clusters corresponding to 1, 2, 3, 2.
Yep.
So would you actually, just considering all this discussion, would you be able to call that this resistant cultivar you were using in your RNA-Seq would be, you could recall them to be RBS1, 2s?
So the original studies for RBS 1, 2, and 3 were identified in the allelism studies. I'm using that same source of RBS3 resistance that they used to identify RBS3. That's the same. A lot of my lines are the same lines that were used in the Rinkner study as well. So did I answer your question?
They identified by, so you said the same lines.
Yep, same lines.
Different disease subtypes to get this 1, 2, 3 separated?
So also for identifying the allelism study?
Yes, yes.
Yeah, so I'm not sure if they use the same susceptible line or not in all 3 of the studies, but they had very similar disease ratings and methods to do those in those experiments.
All right, thank you.